Welcome viewers, welcome to the new lesson from Dr. Arts Biology. If you did not subscribe the channel, you can just click on the subscribe button and bell icon. Welcome students, I hope you are learning biochemistry in a well manner. So we can see that we have already covered the topic carbohydrate under the unit 2. Now we have to move to the metabolism before you forget the carbohydrate structures and all. But metabolism, there is a problem. Now you will be encountering with different types of enzymes and different types of molecules that actually support the chemical reactions happening in metabolism. So first of all, we have to get a clarification or get a clear idea about the different types of enzymes and the different types of energy rich molecules or reducing powers that are involved in the general metabolism. So we will be just covering the enzyme classification different types first, then into the energy rich compound and reducing powers, and after that only we'll be moving into the metabolism. So there is an advantage for that. When you are moving like this, in metabolism, when you encounter different enzymes, you will be familiar with those enzymes. When you encounter different molecules that are supporting the metabolism, you are familiar with that and there will not be any problem. Second thing is, when you are encountering these particular things, you are actually revising the topic that you have covered in this unit. That means enzymes and the energy rich molecules. So there is advantage, that is opposite. One by one you will be learning, as well as you are familiar, and as well as you are getting an opportunity to revise the topic that you have covered. So we are moving in that way. So first of all, we can just cover the topic enzymes. That belongs to the unit 3, and in that unit 3 we will not be covering the entire enzymes now. Instead, we will be just covering what are the different classes of enzymes. Okay, and the remaining topics we will be covering after the unit 2. Hope you understood what I mean. So let us see what is meant by the enzymes and what are the different types of enzymes. So enzymes are nothing but the biocatalyst. So enzymes are biocatalyst. So there is two words in this, bio and catalyst. What is mean by bio is nothing but the relating about a live organism, life. And catalyst is the one which increases the rate of chemical reaction. Okay, so biocatalyst means actually the molecules or substances which increases the rate of chemical reaction that is happening inside a living organism's body. So that's why it is known as the biocatalyst. Now the question is, how this increase in rate of chemical reaction happens? How can enzymes increase the biochemical reactions happening inside an organism's body? So actually it is by decreasing the activation energy. So enzyme increases the rate of reaction by decreasing the activation energy. Now the question is, what is meant by activation energy? Activation energy is nothing but the energy required for a substrate, okay, to reach the transition state. Okay, so it is the energy to reach transition state. Transition state. So energy need to reach the transition state is actually the activation energy. Now the question is, what is meant by the transition state? Is nothing but the state, higher energy state, okay, which is higher than substrate as well as product, and only when the substrate reaches that energy state it can be converted into product. So it is a high energy state in between substrate and the product. If you are just representing in a graph, you can see that there is a substrate, and this substrate will be going up and then it will be coming down, and this is actually the transition state. And the energy level, you can see this one is actually the energy level, so it's comparatively less in case of the substrate when we compare with the transition state, and then it will be releasing energy to form the product. So transition state is nothing but a state between substrate and the product, which is comparatively higher than both. Energy level will be higher than both. That is what actually the transitions. Now we can define enzymes further. Enzymes are biocatalysts which increases the rate of biochemical reactions by reducing the activation energy needed to start the reaction, which are actually mean for the energy required to reach the transition state. So that is actually the enzymes. Okay, so hope you got what is exactly meant by the enzyme. Without enzyme, what will happen? Without enzyme, the rate will be very slow. The living organisms cannot live like this, because enzymes can speed up the reaction up to 10 raised to 16 times faster. It can speed up the reaction up to 10 raised to 16 times faster than the reaction in which the enzymes are absent. So without the enzymes, your life will not be fast like this. You will be very sluggish, because whenever you need a molecule, it will be taking too much time. So if you want to walk, energy is required, and that energy requiring process will be taking too much time, and you will be taking too much time to take a single step. So that is overcome by enzymes activation by increasing the rate of chemical reaction. Okay, now we have to remember this particular fact that in case of the enzymes, there are different types of enzymes that are categorized into 6 main classes. So let us see how the enzymes are classified and who classified the enzymes. Okay, so let us see that particular thing. Okay, so in 1964, the International Union of Biochemistry decided to classify the enzymes so that it is easy for the global community to identify the function and the mechanism of action of a particular enzymes easily. So for that purpose, the IUB, International Union of Biochemistry, appointed an enzyme commission. So EC. So they appointed an enzyme commission to study about enzymes and classify the enzymes into different groups based on their function and their mode of action, etc. As a result, the enzyme commission found there are 6 major classes of enzyme. Remember, we are now dealing with the classification of enzyme, how it was classified and all. So, the Enzyme Commission identified 6 major classes of enzyme, and they have divided this particular thing, divided and subdivided. Okay, they have divided and subdivided this particular 6 classes into subclasses, etc. As a result, an enzyme will be recognized as 4-digit number. So you can identify a particular enzyme with a 4-digit number. In this, first digit will be the major class. It will be belonging to the major class, and the remaining one will be the subclasses. So for your syllabus, you don't have to learn about any of the 4-digit for any of the enzyme. Instead, you have to learn about only 6 major classes. I'm telling this only just to know that how this enzyme classification happened. Okay, how it was done. So 4-digit number will be there for each enzyme by which you can recognize that enzyme, by which you can identify the mode of action, by which you can identify the function of that particular enzyme. Okay, now let us see what are the 6 major classes of enzyme. Okay, the first one is oxidoreductase. The first class is nothing but oxidoreductase. So from the word itself we can see that oxidation is happening. Reductase, what is happening? Reduction is happening. So what is actually mean by the oxidoreductase class of enzyme? It is nothing but the enzyme which catalyzes the reaction where both oxidation and reduction is happening. One of the substrate will be undergoing oxidation and the other substance will be undergoing reduction. That kind of reaction will be catalyzed by the enzyme oxidoreductase. This oxidoreductase is otherwise known as dehydrogenase. Dehydrogenase. Now you have to keep in mind what is actually mean by the reduction, what is actually mean by the oxidation. Some of you may be familiar, but still I am just telling what is mean by the reduction. So reduction is nothing but the addition of H plus or E minus. Electron or H plus acceptance is known as the reduction. Okay. Now what is meant by oxidation? It is actually the removal of H plus or E minus. Is there any other possibility for reduction and oxidation? Yes, it is there. So it is otherwise the addition of oxygen and it is otherwise the removal of oxygen. So these are what actually mean by the oxidation and the reduction. Okay, so the enzyme which catalyzes the reaction in which one of the substrate undergo oxidation and the other substance undergo reduction is known as the oxidoreductase. The substances which undergo oxidation will be functioning as reducing agent. So this particular substance will be acting as the reducing agent. And what about this particular thing? This will be acting as oxidizing agent. So substance which undergo oxidation will be acting as the reducing agent and the substance will undergo reduction will be acting as the oxidizing agent. Okay, so let us see one example. I will be just taking one example which is actually from your metabolism part itself. So let us see the example where pyruvate is converted into reversible reaction. Pyruvate is actually converted into lactate. It is nothing but the lactic acid fermentation that is happening inside your muscle in the absence or in the reduced amount of oxygen. As a result, you will get pain in the muscle. Okay, so that same reaction. It's an example for oxidoreductase. And it is catalyzed by the enzyme lactate dehydrogenase. Lactate dehydrogenase. Lactate dehydrogenase is the enzyme. So lactate dehydrogenase will be catalyzing this particular reaction. Pyruvate is converted into lactate or lactate is converted into pyruvate. Now let us see, there is only one substrate here. We need two substrates for oxidoreductase class of enzymes. So the second substrate will be nothing but the NADH plus H plus. This NADH plus H plus will be donating the proton and electron, and as a result it get oxidized to NAD plus. Okay. So when this particular compound is pyruvate is converted into lactate, pyruvate undergo reduction, and the reducing agent will be the one which is undergoing oxidation. So it is undergoing reduction, as a result it is oxidizing agent. So it will be oxidizing which molecule? Nothing but the NADH plus H plus. NADH plus H plus undergo oxidation. So it will act as a reducing agent, the one which gives electron and proton to the pyruvate. So as a result, you can just see the lactate is formed. Now let us take the reverse reaction. Here lactate is in reduced state, because you can see that in the reaction pyruvate get electron and hydrogen, as a result lactate is formed. So lactate is in reduced state. In this lactate will be converted into pyruvate by the same opposite reaction. NAD plus will be accepting hydrogen and electron from the lactate, and it will be getting reduced to NADH plus H plus. We will be seeing why we are writing NADH plus H plus and all when we are discussing the high energy molecule and reducing powers after this particular enzyme part. So okay, so this will be undergoing reduction. Now in the opposite reaction, who is the oxidation agent, the oxidizing agent, and who is the reducing agent? This one is reducing agent and this one is oxidizing agent. This one is undergoing oxidation and this one is undergoing reduction. Okay, so in the forward reaction where pyruvate is converted into lactate, pyruvate is the oxidizing agent and pyruvate undergo reduction. NADH plus H plus is the reducing agent and it undergo oxidation to form the NAD plus. So pyruvate is converted into or reduced to lactate. In the reverse reaction, lactate is converted into pyruvate, where lactate is a reducing agent and it undergo oxidation to form the pyruvate, and NAD plus is the oxidizing agent. It undergo reduction to form the NADH plus H plus. So let us see the reaction then. Okay, so we should know the structure of pyruvate. We will be seeing the structure in glycolysis also. It will be easier for you. I am just taking the example from the portions that you are learning as per your syllabus only. So pyruvate is a keto acid. You can find COO minus, C double bond O, CH3. Okay, and this is undergoing oxidoreduction by the enzyme lactate dehydrogenase, and you can find that it will be forming lactate, COO minus, CO, CH3, where we have to add hydrogen. It is at the second carbon. You can see here the hydrogen will be added in this way. So lactate is in reduced state. This is undergoing oxidation, reduction to form the lactate. So this one is a pyruvate and this one is lactate. Now from where this hydrogen comes, we have already discussed. It is nothing from other than the NADH plus H plus is undergoing oxidation. As a result, the NAD plus is formed. And this 2H is actually added in these points. In the opposite reaction, the lactate is acting as a reducing agent undergoing oxidation. As a result, this 2H will be coming to the NAD plus and it will be forming NADH plus H plus. And as a result, pyruvate will be formed. And this enzyme is actually catalyzed by the enzyme lactate dehydrogenase. Okay, so LDH will be catalyzing this particular enzyme. So that's about oxidoreductase. Now let us move to the second class of enzyme. when you study the enzyme classes, you have to keep in mind that you have to learn in the exact order, because each one is actually represented by a number. You have already learned the 4-digit identification number will be there in case of the enzyme, and the first number is nothing but the enzyme class, major class. So you have to remember the enzyme class in order itself. Okay, so to remember the order, you can just find out any of the keyword. Okay, I am just using this one, oath lil. Okay, oath lil. It should be the order in which you are writing the enzyme classes, because the number is also a matter. Okay, so oath lil is the one which I will be using. Now we can see that the second one, the T. So the second class of enzyme is transferase. Transferase enzyme. Okay, so this transferase enzyme, from the name itself we can see that it is actually transferring. It will be transferring something from one substrate to another. Okay, so they are actually group transfer. They are catalyzing group transfer reactions. They are actually catalyzing group transfer reaction. That means from one group to another, the specific group will be transferred. For example, phosphate, sulfate, etc. will be taken from one substrate and it will be added into the next substrate. That kind of reaction is catalyzing enzyme is known as the transferase. So transferase catalyzes the reaction where the group transfer occurs between substrate. These are the enzyme which catalyzes the group transfer reactions. Okay, let us see one example. In your glycolysis, you have glucose. So you have glucose, which is actually irreversibly converted into glucose 6-phosphate. So glucose is irreversibly converted into glucose 6-phosphate. Okay, so in that reaction, why this particular thing is happening? So let us see that the transferase enzyme that is actually catalyzing the glucose to glucose 6-phosphate. What happened? You can see that one phosphate group is added to the glucose at the 6th carbon position. So transferring happened. So what is the enzyme? The enzyme that catalyzes this particular reaction is nothing but the hexokinase. So hexokinase belong to the enzyme class transferase. So as a result, glucose is converted into glucose 6-phosphate. So who will be the donor of this phosphate group? That comes the question. So it is nothing but the ATP molecule. So ATP will be converted into ADP. We know that ATP means adenosine triphosphate, that is converted into diphosphate. So one phosphate is removed. So that one phosphate is added to the glucose 6th carbon position. As a result, the glucose 6-phosphate is formed. So this is the example for transferase class of enzyme catalyzing reaction. Example for this enzyme is nothing but the hexokinase. You will be learning lot of enzymes under each class when you are covering the topic metabolism of different macromolecules. Okay, so let us see the structural reaction here. You can find the glucose structure. You are already remembering the glucose structure because we have learned the carbohydrate already. Okay, so glucose is having HOH, HOH, OH, HOH. Hope you remember this structure. Okay, you just pause the video and draw it yourself so that you can ensure that you are remembering this particular things which you OH. Okay, now it is with the ATP, adenosine triphosphate. You will be learning the structure of adenosine triphosphate in energy rich molecule part of your chapter. Okay, Now this particular ATP, I'm just writing ATP with the red one so that you can understand it is coming from the ATP. Okay, ATP. Now as a result, what we can see, glucose 6-phosphate should form, and at the same time it should release ATP. ADP should be released out. So one phosphate group is added to the glucose to the 6th position. So we can just see that here. Okay, so this is the 6th position, CH2OPO. Okay, so we can see that the phosphate group is, we have already learned about phosphate group P, double bond O, OH, OH, OH. Okay, and when you are adding this phosphate to this particular thing, one water molecule should be removed. We know that CH2OH is there. Okay, and this OH should be removed plus one of the H from here. Okay, so as a result, CH2OPO, O is already here, then 1, 2, 3, PO3. How many minus? 2 minus will be there because this hydrogen and this hydrogen will be released out. Okay, so OPO3, 2 minus, phosphate group is added, and remaining portion will be exactly same as what we have drawn here. Okay, H, OH, H, OH, OH, H, H, OH, H. So this is actually glucose, and this one is glucose 6-phosphate. This is the enzyme catalyzing. This is a reaction catalyzed by the enzyme transferase class. Example is the hexokinase. Okay, this session I am just stopping here because it will be too long to upload in the YouTube. For the convenience, I will be just stopping here. So you have learned just 2 classes of enzyme, and the remaining 4 classes of enzyme will be just dealing in the next part. So you can consider this as the part A of unit 2, unit 2 topic 1 part A. Okay, and then part B can be uploaded in the next. You have to watch that particular video in order to completely learn about the entire classes of enzyme. So remaining 4 classes I will be just dealing in the next video, that is unit 2 topic 1 part